my great pleasure to introduce Matthias Stuber, who is the CIBM MRI CHUV UNIL Translational MR Imaging Section head, and he will be chairing the session today, as well as be giving a little introduction on our speaker, who is Christopher Roy. Thank you very much.
Thank you very much, Tina. Good morning, everybody. I'm clearly the less interesting part of this meeting this morning, but I'm here. My job is to introduce Chris Roy, who I'm sure will be giving an outstanding presentation. Now, speaking about technical difficulties. All right. So this is Chris Roy in a nutshell, as you can see, right? So Chris did his PhD in medical biophysics in Toronto, in Canada, with Chris McGowan at SickKids. He then did a first kind of a abbreviated postdoc at the same place, I guess, from 2017 to 2018, before he then joined us working on one of my SNF grants focused on cardiovascular imaging, which he already did in Toronto, because he was focusing on imaging of the fetal heart. Now, he then also became a junior fellow of the ISMRM, and you saw that on the first slide. This is a very prestigious award. If you're a young scientist who is very accomplished, you may apply for a junior fellowship, and many of you who are also doing MRI and are junior, so that excludes myself, for instance, could apply for this distinction, and I encourage you to do that. And then he became a Maître assistant as a principal investigator on an Ambizione grant, where he actually took all he's learned at the CHUV and went back to his first love, which is fetal MRI. Just to show you a bunch of pictures of Chris Roy, here he's proudly presenting at the SEMR in 2020 in Orlando. This was the last in-person meeting before COVID really hit hard. Here he's presenting at our annual retreat at the Lac d'Annecy. Here, that's a very nice picture because it shows him with his former mentor, who initiated this entire field, I would say, even of fetal cardiovascular MRI. That's Chris McGowan on the right-hand side. On the left-hand side, it's the pioneer of 4D flow imaging. That's Michael Markle. In the middle, of course, Chris Roy, with a bunch of beers. So this is a very nice photo. Then here, I'm not sure how serious he is with playing this baby foot here, just one-handed. But I think it's enough that's in my backyard during the annual group barbecue. Here you show him with his lovely family at the ski weekend this past, I think, January, and finally, again, you know, with his awesome Junior Fellow Award of the ISMRM. Now, we are doing cardiovascular MR. Cardiovascular MR is hard. Why? Because the heart is constantly moving, because it contracts and relaxes in a living human being, right? On the one hand, but there is also respiration, and there are small anatomical structures we would like to visualize. So the heart is actually a tough problem. So what Chris did, and going back to the nutshell, he decided now to image the fetus's heart, right? This is even smaller, it beats even faster, and it's in the middle of a much larger object, which is the mother. So and on top of that, you cannot even get an EKG out of that to synchronize the acquisition to the rhythm of the heart. So it's basically a crazy task to pull this off, to visualize the beating heart of a fetus with MRI. And why did I show this knot? The size of a fetal heart is like this little knot there. So that's about the size of the fetal heart. So it's crazy to go about this. It's crazy to try to do this, but Chris did it. And now to the more interesting part. Chris, please show us whether you are capable of doing this or whether it stays a crazy idea.
Perfect. Thank you very much, Matthias. Okay, well, welcome everyone. It's great to be speaking in front of people in person again. And so Matthias just hit on a lot of the main bullet points that we'll go through in this presentation. And as he said, my interest dating back to the beginning of my graduate work in Toronto and Canada has been to image the fetal heart. And so here we have a wonderful real-time MRI video of the fetus moving in utero. So the brain is up here. You can see obviously going into the abdomen, the heart. You can see the limbs moving around. And this is a really challenging, or using Matthias's words, crazy expedition to try and image the anatomy of the fetus in utero. But I'm hoping that through this presentation, we'll understand the whys, why we want to do this, and why it might not be that crazy based on the technology that's been developed within the community, but especially in Matthias's lab and work that I plan to do as part of my Ambizione grant. So let's talk about motion. So as you all know, in any type of imaging, motion can be a problem. So here we have, just with a cell phone camera, trying to take a picture of my dog. And sometimes he sits still, and we have very nice, well-detailed features. But a lot of the time he moves. And not only is he no longer in the position that I want to see him in, but all of his fur is completely blurry. And this is a pretty good analog for trying to image the fetus. It's not just about the fact that the image quality can be degraded. It's that we might not be imaging the part that we want to. But of course, motion can also be a really great source in imaging. So if we think of the why of why do we want to do cardiac MRI and why do we want to do this in the fetus, it's because we have quantitative imaging that makes use of this motion. So if you think of imaging the heart in 3D, it's also dynamic. We can visualize the vessels, we can visualize the function in a truly comprehensive way. Not to mention, if then you add quantitative flow on top of this, we really have powerful tools that are useful for imaging the heart, not only postnatally, but as I hope to show you, in prenatal life as well. So in the talk, we'll go over some of the real key motivation factors for why we want to do this type of imaging, how might it be useful in a clinical setting, but also why this is challenging. So as Matthias alluded to, fetal cardiovascular MRI is very difficult. The size is very small, the things are moving quickly, and we'll talk about how this impacts our imaging. I'll then go over some of the work that I did in Toronto during my graduate work, starting with 2D imaging of the fetal heart. As Matthias mentioned, my move to CHUV, which was really a focus on 3D imaging of both the adult and pediatric heart, and then how this all ties together to go back to my first love, which is fetal imaging, and using the methods that I learned for 3D imaging to really take this to the next level. So the work's motivated by congenital heart disease. And so congenital heart disease is the most common congenital abnormality. It's about 1% of live births each year. Roughly 700 children born in Switzerland each year will have a congenital heart defect. And there's really a range of possible defects that this is covering. But overall, they're structural abnormalities. They're either issues with the chambers or the vessels of the heart. And so if we compare the normal heart here on the left with its 4 chambers and major vessels, you can see that some defects could just be small holes in between the chambers. Now, depending on the severity of these, they can be quite small and actually just resolve on their own. And so this doesn't really require any type of intervention, and many people have and live with these small defects really without knowing it. So I'm just going to make sure great. But as these things get more complex, as we have narrowings of vessels, as we have potentially swaps in between how the vessels are connected to the chambers, this ultimately impacts the way that in prenatal life, the oxygenated blood is getting to the developing organs. So this can actually affect development, but also in postnatal life, once the child is born, if these are not corrected, if they remain sort of misconnections in between vessels and chambers, this can prevent oxygenated blood from flowing through the body, and this really requires serious intervention. So where does imaging come into play? Imaging is really key all the way from first screening and finding these defects in prenatal life, and then all the way through adolescence and into adulthood, and trying to monitor potential interventions that need to be done. And overall, it's been shown that prenatal diagnosis really does improve outcome. So it's important to have as much information as we can from the very beginning of these congenital abnormalities. And I think most of us are familiar with ultrasound being the main driver of imaging in the fetal life. And again, as I said, all of this is essentially to be able to have the best patient management that we can, so that we can monitor a really increasing list of different interventions that exist in prenatal life, as well as continuing on into adolescence and adulthood. So if ultrasound is really the primary imaging modality, why would we want to use MRI? Well, there's been a really growing interest in the use of MRI to image the fetus. In terms of complementing ultrasound, this typically occurs in a couple cases where you have, for example, low amniotic fluid can really prevent the ultrasound transducer from getting a good window to the fetus. Similarly, you have the same problem with maternal obesity, but also just in general during late gestation as the fetal bones grow, this can really make it difficult to assess the complex anatomy, especially in something like fetal congenital heart disease. Now with MRI, we really have, as I showed you in those earlier slides, quite a comprehensive way of imaging the heart. We can look at structure and anatomy, we can measure things like blood flow, and with things like tissue characterization, we can actually quantify things like the transport of oxygen. And so really there's been a growing number of groups and researchers that have been looking to MRI to complement ultrasound and provide something that's much more comprehensive in the way that we assess the fetal heart. But this is of course not without its challenges. So coming back to this crazy idea of imaging the fetal heart, on the left here you have sort of a typical 2D dynamic image of the adult heart. So you have the left ventricle and the right ventricle, and this is a representative single heartbeat that we reconstruct from data over several seconds while holding our breath. If we then compare this to the fetal heart, the first thing you'll notice is it's obviously much smaller. So here we have the left ventricle, right ventricle. You can see the relative size differences, but also it's beating almost twice as fast. And so what this means in terms of our imaging problem is we need better resolution to be able to identify the smaller structures in space, but we also need better resolution in the frame rate that we image at so that we can pick up this increased fetal heart rate. So it's small, moves fast. And then as Matthias alluded to, we don't have an ECG. So typically in this type of adult imaging you see on the right, we have to image over many heartbeats, and then we create one representative heartbeat that is at high resolution, both spatial and temporal. And this is done by combining information from the same part of the cardiac cycle because we're synchronized to this ECG signal based on electrodes that we hook up to the body. So one problem is we don't have that in the fetus, and we'll come back to how we actually solve that in a little bit. But on top of all of this, we have unpredictable movement. So coming back to the first slide, we have a fetus that's moving around. So even if we do manage to somehow synchronize our heart rate and have the right spatial and temporal resolution, we have to do everything fast enough so that unpredictable movement doesn't impact our scanning. And so the solution that I began working on several years ago in Toronto, and other groups have certainly taken the same approach, is really to have flexibility in our MRI acquisitions. So here we have two different ways that we can image the heart. We have real-time images on the left and cine on the right. And the real-time it's not real-time in the sense that this is what we see on the scanner, but it's that we can collect our data, and then after the fact, we can chop our acquisition into basically small blocks of time on the order of tens to hundreds of milliseconds. And this allows us to really see the motion as it occurred during the scan. And this is really important in the fetus because if we then want to put together the same cardiac positions, we need to be able to have a way to see the motion and either correct or compensate for it. And so here we have, again, this image of the fetus moving here. And if we look at the temporal signal going through the image horizontally and we look at it over time, you can see that we can very well monitor maternal respiration. And we can even visualize things like gross fetal movement at the end. And what this will allow us to do is to implement some motion compensation strategies. So here we have another example of two real-time reconstructions of the fetal heart. And here we're actually able to track the displacement of the heart through time, which we can then in turn use to stabilize this section of the image. And here again, showing both a minor displacement, so not that much motion, but then here really considerate displacement of the heart due to maternal respiration. So while that's useful, we can stabilize the position of the heart, we still need to be able to combine different phases of the heart without having an ECG. And so one approach to doing that is something we developed in Toronto called metric optimized gating. And the idea here is that although we don't have the ECG, we can start with a model, or a better word for it, really a guess of what the ECG would have been. And when you have a guess that's poor, you can see that this image here, it's not pulsating, it's not moving the way it's supposed to for a dynamic image. But as we slowly iterate through better and better estimations of what the underlying cardiac cycles were, you can see we get more and more pulsatility, we get something that's much more dynamic. And this can all be quantified by a metric that's basically telling us whether we have this motion blurred image or whether we have the dynamic image that we have at the end. And so iterating over this, we can actually retrospectively replace the information we would have had from an ECG. So with that, to recap, we have motion correction. We can stabilize the position of the heart. We can combine the different phases of the cardiac cycle, despite the fact that we don't have the ECG. But then finally, all of this we're trying to do very quickly. So we're not collecting that much data. And additionally, if we have these large periods of gross movement, we may need to reject things that even cuts down the acquisition even shorter. So then our last step is we have to have a type of image reconstruction that will allow us to deal with the fact that we have heavily undersampled data. And so many of you might be familiar with the idea of compressed sensing, but we'll just quickly go over how it applies here in 2D fetal imaging. So if you think of the dynamic fetal heart here, that is fully sampled. You can see it's quite nice. We have strong details and we don't have much artifact. If we have to reject a lot of the data, or if we have to do an acquisition that's much faster, we end up with this undersampling artifact. And so the basic idea with compressed sensing is that we have some representation of our dynamic image that's going to allow us to separate this artifact from the features that we care about. So here in this case, we're taking the Fourier transform along the temporal domain. And what you end up having is the DC component, so the average motion of the image. And then in the fully sampled, you can very nicely see the different frequencies of cardiac motion. In the undersampled case, we again have a nice DC, and we have the components of the cardiac motion that are just sort of slightly above the artifact level. And so we have an iterative reconstruction that is basically trying to minimize the signal in this Fourier transform domain, because we know that, especially in the frames where we have the cardiac motion, it should be relatively sparse. Most of these pixels should be low values with a small collection of high ones. And as you can see, as we iterate through this reconstruction, we get rid of the artifact, and then the resulting image that you see here on the left becomes closer and closer to what we would have had if we had sampled for the correct amount of time. So altogether, we have ways to track the motion in 2 dimensions. We can combine different parts of the cardiac cycle, and then we can even deal with the fact that we haven't collected enough data. And so here we have some examples of what this type of imaging looks like when we put all of these different pieces together. So here we have short axis images. So you have the left ventricle and the right ventricle, both before and after motion correction. If you recall the slide from earlier, this corresponds to the minor movement case. So as you can see, there's not really that much difference between the image with and without stabilizing the underlying motion. And for the same volunteer in the 4-chamber view, where you have the left ventricle, left atrium, right ventricle, again, you see the same thing. Not much change when we do motion correction. However, in the much more significant motion case, where we really had movement of the heart due to maternal respiration, you can see that without motion correction, it's very difficult to make out the features of the heart at all. And then once we add this motion correction, we have a much more dynamic and a much more sharp visualization of the cardiac features. So all of these methods together really created this framework that we had for imaging the fetus, which would then allow us to start actually looking at congenital heart defects. So here we have a series of images from the same fetus. So this is a stack of images that is covering the heart. And I'll just quickly explain how these images are used to actually try and identify congenital heart defects. So very quickly, basically what you're doing is you're tracking the position of either the chambers or the vessels and seeing how their connections go. So for the right ventricle, we can assess that this is in fact the right ventricle due to this structure called the moderator band. And if we follow it through each of these images, we see where it starts to connecting to a vessel. And if we continue to follow that vessel, we see that that vessel that's connected to the right ventricle is connected to the head and neck vessels. Why is this important? This means, by virtue of connecting to the head and neck vessels, that it's the aorta, but it's connected to the right ventricle, which it should not be. The aorta should be connected to the left ventricle, and the main pulmonary artery, which we can see as this bifurcating vessel, should be connected to the left. So it's this type of connection between the chambers and vessels through multiple images that we need to properly assess the anatomy to have some clinical utility. So with that in mind, let's look Just briefly, this was a paper that we did looking at 30 fetuses with this type of technique. And what we found was overall, by comparing MRI to ultrasound, we had lower identification of these congenital heart defects. So not as good an ability to connect these different structures together. However, if we looked into the data and sort of looked at the subgroups between whether we actually properly covered the heart or not, if we covered the heart with MRI, we did just as well as ultrasound. And together, these two modalities actually gave us a better identification rate than either one on its own. So there's certainly promise here, but there was kind of this nagging question of why are we not getting coverage of the heart? And so here, let's come back to the idea of real-time images. This is, again, supposed to be a stack that's covering the entire heart, and I've sped up the real-time images so you can kind of see this, pinball of the fetus around. Now, if we look at the cine images that come out of these real-time images, so this is using that whole framework for motion correction that I described, we still get really good image quality for the individual two-dimensional images. The problem, however, is that the positions that we initially started with, so we wanted to cover the entire heart, that is not necessarily still the case. And so we actually have multiple instances of essentially the same slice position as the fetus kind of moved in and out of frame. And if you compare this to something like ultrasound, which can really sweep through the anatomy, this becomes a problem where we can't connect our two-dimensional images and can't visualize the structures across many two-dimensional images. And so this really comes back to our problem of motion. We can deal with motion within individual images, but when we try and connect them together, it really becomes a problem. And so as you see, we have the heart, we're trying to cover the structures with these two-dimensional slices, but this type of motion really results in, yes, good individual images, but cross-sections that just don't represent what we need clinically. And so the result of all this work, which really was fundamental in showing that we could even use MRI to image the heart, was that, yes, we can do things really well in 2D, but we're still trying to solve a 3D problem with 2D imaging. And so this really led me to the conclusion that 3D imaging is where we need to go next. So I thought of that on a Friday afternoon. Saturday, I just got on a plane and I said, I'll go to Switzerland. They really know how to do good 3D imaging in Professor Stuber's lab. Not quite the case, but pretty close. Professor Stuber's lab are really pioneers in 3D imaging of the heart. And as you'll see in the next section of the talk, I was really motivated to come here and learn, and to some extent leave the fetal imaging behind and learn what's difficult about 3D imaging just for adults and pediatrics, as we talked about earlier. And so this really was a great change in both my research direction, but also added new opportunities, new challenges. So we're going to switch gears a little bit. We're going to talk about conventional 3D MRI. So we already talked about 2D MRI, trying to create these representative dynamic 2D images. Now we'll talk about 3D. And if you're just interested in the structures in the 3D heart, really the most conventional way to get rid of motion is ECG triggering and respiratory navigation. So what does that mean? It means that we're really limiting where we acquire the data. We know where we are in the cardiac cycle, or at least we think we do, and we only collect data when we're in a specific part of the cardiac phase, and we only collect data when we're in a specific part of the respiratory cycle. And this is trying to freeze motion. And this works very well. However, it can lead to really inefficient, but more importantly, unpredictable scan times. Because as you can imagine, if you breathe differently between the start and the beginning and the end of the scan, if your heart rate goes up or down, it's going to completely change how this data is collected. So one concept to try and improve the imaging efficiency is dealing with the respiration in a different way. And so instead of limiting our acquisition to just certain parts of the respiratory cycle, we can sort of image independent of where we are in the respiratory cycle, and then we'll deal with stabilizing this motion, just like we were talking about earlier for fetal imaging, after the fact. And so one concept of this is to basically have a repeated part of your MRI imaging data that is going to tell us something about the respiratory motion. And this type of approach, which was really, I think, well pioneered for radial imaging, Professor Stuber's group, has been really outstanding at providing high quality images with an improved scanning efficiency. So we have the benefits of freezing motion, but because the respiratory, we never stop during the respiratory cycle, we have a better efficiency in the data we collect. And in previous works, the way that we actually correct the respiratory motion, because we are scanning throughout the whole cycle, has been an approach to essentially correct the main direction of the respiratory motion, which is usually the head to foot or SI direction. Alternatively, we don't have to correct it. We could actually create images at different respiratory positions. And this is another way that we can do it. And while this is a strong approach, in order to do so, we end up having undersampled data for these different positions. So we have to use a compressed sensing algorithm like I showed in previous slides. And so one idea I had when I came to Professor Stuber's lab was that I think we can improve this idea of respiratory motion correction. And it kind of brought me back to the same type of approach that I used for synchronizing the fetal acquisitions in terms of the heart rate. And this is this idea of essentially having a model that describes our motion that allows us to put the images together in a different way, a metric that tells us whether the images are getting better or worse, and an iterative algorithm that will improve things. So just very quickly, in this case, we know that from this repeated part of imaging data, we have a signal that is very well correlated with our respiratory motion. What we don't know is how much motion does that actually mean in a physical unit in our three directions. So we take our model of motion that comes out of this repeated imaging data, as I said, and then we build this assumption that this signal is proportional to our respiration if we multiply it by some coefficients that will then give us physical units for motion. And so if we think of how this actually works in practice, so here we have some simulated data that we're going to use to demonstrate this. So I've created essentially synthetic MRI data with a respiratory motion. And as you can see, this image is relatively blurry. But we can get our respiratory signal out of it, and we can begin to have estimates of the three dimensions of respiratory motion based on this model that we've created. And using a metric for image blur, we can see whether or not our model's getting closer and closer to the actual true underlying physiological motion, with the assumption being if we get this motion correct, we'll have the sharpest possible image. And so as you can see, as we iterate and we converge to our sort of minimum solution here, so this representing the least amount of blur, we have an image that went from being blurred due to respiration to being quite sharp because we've corrected that motion. And so we applied this in a number of different ways. First, using the same type of numerical simulation that I just used to demonstrate the technique. And we compared it to the previously described one-dimensional correction of the data, the respiratory resolve for reconstruction of the data, and then this is the proposed method. And so what you see here in each of these rows is simulated data with different levels of respiratory motion. And so you can see at low levels of respiratory motion, we still visualize some structures of the heart, but they're certainly blurred. And then as you go to larger and larger amounts of respiratory motion, really becomes difficult to assess anything in terms of structure. Similarly, if we have just a one-dimensional correction of this motion, we do pretty well at low levels of respiration, but this starts to break down as the motion gets more complex. If you then compare either the respiratory resolved or the proposed correction for 3D motion here, we really, across the different levels of respiration, have something that is quite similar to ground truth, albeit with the proposed method, we can reconstruct our images much faster. So we applied this. Also, I had the luxury of, as I mentioned before, the acquisition method for this data was really pioneered in our group, and I was fortunate to have literally thousands of data sets that I could go back and look at and apply these algorithms to. And so we looked at a cohort of volunteers and a cohort of patients, and here are just a couple of representative examples. Again, showing the effects of not correcting respiratory motion, the simple one-dimensional correction, the respiratory resolved, and then the proposed 3D respiratory motion correction. And overall, what I hope you appreciate is whether we're looking at the right coronary artery, or the left anterior descending, or the left circumflex. In all cases, we really improve the visibility of the entire length of the vessel using this 3D correction. And so just quickly, We also have several quantitative metrics that we use to really corroborate what you're seeing by those images, not only in determining whether we actually estimated the correct respiratory motion. So in our simulated case, we could actually look at what did we put in for respiration, what did we get out, and overall we had good accuracy there. We saw using quantitative metrics for image sharpness that overall our method produced significantly sharper coronary arteries. We also had two experts, so Matthias and Davide Piccini, go through and choose the best image in all of our patient data sets. And this resulted in always choosing our proposed 3D motion correction in vast majority of the data sets that we looked at. And then finally, and then an additional scoring of the images by experts, again, showed significantly higher image quality. But then finally, as I showed you in some of the images, we had similar image quality between the respiratory resolve and the 3D motion correction, but the reconstruction time was much faster. So it's a much more efficient way of reconstructing the data. So the takeaway from this slide is, yes, we've come up with this method of dealing with respiratory motion in a really efficient way. But I think what's maybe most interesting from my perspective is that we built this method for correcting respiratory motion using something that's very simple, this one-dimensional signal to respiratory motion. And what we get out of it is something very powerful. And in the following slides, we'll see where this is useful because you kind of almost get this correction for free in this type of imaging. You always have this repeated sample, and I'm going to show you how we can use that in other ways to have high-quality imaging data. But first, before we get there, just want to recap. So we have a method for correcting respiratory motion correction, and this improves our scanning efficiency because we're not stopping during different parts of the respiratory cycle. But we're still limited in terms of our efficiency by this ECG triggering. So the fact that we only collect imaging data during certain parts of the cardiac cycle, again, means that we're at the mercy of different changes in the heart rate, changing our total scan time. And so another concept that was really pioneered by Professor Stuber's group in Lausanne is this idea of free-running imaging. So now we're really not only abandoning stopping because of where we are in the respiratory cycle, but we're also abandoning stopping doing where we are in the cardiac cycle. And what this means is we're really continuously acquiring MRI data in a very efficient manner. And in the same way that we were able to get a signal that represents our respiratory motion, we can actually get a signal that represents the cardiac motion as well. And we will use this to put this data back together in different cardiac and respiratory phases. And so if you see here, here are some representative reconstructions of this type of imaging. So because we've never stopped sampling, we can actually look at the different motions that exist. So we have the cardiac motion, which is dynamic here, but we could also look at the respiratory motion, which is also dynamic. And as I said, this is really an efficient way of acquiring our data because we don't have to be subject to changes in respiratory or cardiac cycles. We just collect for a fixed period of time, and then we use these motion signals to put things back together. And really, one of the main benefits that I saw in this is that we have a high potential for using this for imaging congenital heart disease. If, again, we come back to the idea that we want to connect these different structures and see what their impact is going to be, having a type of imaging where we just really put a box around the heart, we don't need to worry about interruptions in our sequence, and we just collect all this information. This is potentially very useful for assessing congenital heart disease. But the problem of motion comes back. So, especially because we're trying to assess congenital heart disease in pediatric cohorts, the challenge of motion is going to exist because unless you either sedate or you put these children under general anesthetic, chances are they might move during the 5 or 10 minutes that we acquire these data. Now, what's nice though is because we have this acquisition, this free-running acquisition that continually samples, I just showed you how we can look at the cardiac and respiratory motion, But we can also revisit the idea of a real-time reconstruction of these exact same data. And that's what we're seeing here. You're seeing 3D volumes that are sequentially reconstructed after the fact, and it's making it quite clear that this patient was moving throughout the scan. And if we think of what this then does to our image quality, here we've used our cardiac signals to try and get back the cardiac motion. And truth be told, it does a fairly reasonable job despite the fact that there's all this motion. But I think you can appreciate that the overall image quality is really brought down, and this is due to the fact that the patient just moved the entire time. And so, yes, sedation and general anesthesia would be solutions to this problem, but what if there's another way that we could do it? And so we come back to really the same place we were with fetal imaging, which is we want to take advantage of the flexibility of this data in our reconstructions to use these real-time images to stabilize our motion and hopefully improve our cine images. So here we have another example of an even a more severe case of motion throughout the scan. So this is, I think, about 5 minutes of scan time, and you can see that the subject is really just moving the entire time. So we have two problems. We need to try and find a way to stabilize this movement, but also remember that they're breathing the entire time. And one challenge that we have in pediatric imaging that was less so a problem in fetal imaging is that it's quite difficult to decouple this bulk motion from respiratory movement. And this is where FNAV, this 3D respiratory motion correction that I introduced in the previous slides, now comes back into play. Because we essentially get this estimated respiratory motion within our data anyway, within these individual real-time 3D volumes, we can correct the respiratory motion and remove that as a compounding factor. And so what we're left with is the ability to try and track the position of the heart through these real-time 3D images. We can stabilize this using 3D rigid registration, and in the cases where there's really just too much motion to register, we can apply outlier rejection. And then when we put this all together, we have much improved image quality. So here we have, again, these are cine images. This is using our cardiac motion information to resolve the cardiac cycle. And this is the same thing, but with the motion compensation provided by our real-time 3D imaging volumes. And I think you can appreciate that we really get very nice image quality, both in the terms of the dynamics that we can resolve and the individual structures, despite the fact that this patient was moving throughout the entire scan. And so this is really preliminary work, but we've started validating it, again, coming back to a numerical simulation where we can control the amount of motion that we put in and see what effect this has on our reconstructions. So with very low amounts of simulated patient movement, we basically have the same type of image quality whether we correct or don't. As we start to have more moderate motion, it's very clear that the structures become blurred, but we can recover this image quality with our motion correction strategies. And then even to really severe bulk movement, the combination of our motion compensation really allows us to still have quite nice image quality, which in each of these cases compares very well to the situation we would have if there was no motion at all. So then just a couple more examples in patients. So again, here we have slightly less severe motion, just sort of some movement side to side through the scan, but it does still have quite a large impact on the overall image quality, but we can essentially get back this image quality with the motion compensation. And then finally, again, this is an example we showed earlier, but now showing the motion compensated version. We really are able to maintain high image quality despite these movements throughout our acquisition. And then we've, again, like I said, this is preliminary data, but just looking at scoring of these images, which in this case was actually done with an AI algorithm that was trained to provide an assessment of image quality for this type of data. You can see that overall our corrected images, which is the 40 images here, compared to the uncorrected images, we have really better image quality across the board, and can in fact even take images that would otherwise be of little diagnostic value and bring them to at least a moderate diagnostic value. So overall here we have a framework now for doing 3D imaging of the heart, even if there's bulk patient movement. We have very strong preliminary results. We're still in the process of validating this, both the numerical phantom, but also in volunteers where we can give them instructed movements and see what impact this has on image quality. But overall, the importance of this work is that with this way of imaging the heart in 3D, despite the presence of motion, we can really look at decreasing the need for sedation and general anesthetic, which could be really beneficial in pediatric populations. But now we return to the fetus. So with this framework that we've developed for, imaging the heart continuously without having to stop or have any a priori knowledge about the motion, I now really see this as something we can go back and look at the fetal heart for and try and bridge this gap between high quality 2D images that don't provide enough coverage and instead basically putting a box around the fetus and having all of that information in there, which if we don't correct is admittedly going to be quite blurry. But if we look at our real-time 3D images and track the motion, the perhaps optimistic goal is that we will correct this in such a way that we will bring back really high quality imaging. But it's not just hopeful. We actually have some preliminary data to show that we think this might work. So here we have real-time 3D images of the fetus. And in this case, you can see there's sort of stability for some part of the scan and then sort of a movement that occurs. And in our preliminary investigation, we've just been focusing on trying to stabilize the brain. Even though our ultimate target is the heart, we thought we'd at least see if we can take this larger non-deformable structure and see if we can co-register it in such a way that we can improve the image quality. So here we're just showing taking these 3D images, focusing on the brain and trying to stabilize the motion. And what's really cool is if you think of the original reconstruction where we don't do any compensation, the fact that the fetus is moving throughout the scan means we really blur the structures overall, and especially if you try and see any fine detail in the brain, this is really blurred. If we apply our initial motion correction algorithms, we really already recover a lot of fine features. And for those of you that image the brain, I'm sure you'll look at this and say, well, this is not the contrast that you want for brain imaging. Very aware of that. This is really just a preliminary look using the same sequence we do for cardiac imaging. But hopefully you'll appreciate if you then focus on this next example, this really is providing us a continuous isotropic scroll through the heart, through the brain in this instance. And if we can get to the same point with the heart, we've really solved this challenge of having motion during our scan, but still being able to connect features, vessels and chambers in a way that allows us to assess these congenital abnormalities. And so here, this is our first attempt at trying to do the same thing for the heart. We're not dynamic yet. We're just trying to stabilize the motion and see if we can visualize the structures. There's still some work to go on improving the contrast, the overall noise level. But it's really quite heartening that we can have this isotropic resolution that allows us to really arbitrarily now go back and interrogate the different slices. So we don't miss any of the slices, even if the fetus moves, because this imaging box encompasses the entire body. So unlike our previous 2D examples, where we would sometimes not see the features that we were looking for, this is really the idea here is to ensure that we can. So just to end, I'll just talk about some of the next steps. So this project now for imaging the fetal heart with 3D, as Matthias alluded to, is part of an SNSF Ambizione grant that I just started last month. So we had a PhD student that just started, and now we're really getting into the validation and the optimization of this. So we need to look at how can we get the best signal out of the heart with this sequence, try and do this with minimum artifacts, shortest scan time possible. And this is really going to be done through sort of a comprehensive test. So much like I showed for the adult imaging, we have a similar computer simulation that allows us to put in respiratory motion, bulk fetal movement, and really test our algorithms in a way that's controlled and gives us a ground truth. We also have a motorized phantom setup that the PhD student that just started, Robin, has already began working with. So he's trying to build in some more complex motions compared to sort of the more periodic motion that we've used this phantom for in the past. And then with that, we'll move into adult volunteers and then eventually pregnant scanning. But ultimately, the goal here is to take all of this, robust imaging and provide fetal imaging with the same type of comprehensive tools that we have for pediatric and adult imaging. So really being able to interrogate the structures, without losing any slice positions. But it extends beyond that. Adding dynamics, we can look at really trying to quantify the ventricular function, and then adding flow sensitivity, for example, we can provide some quantitative evaluation as well. And so we have a long path to get there. And then once we do, we'll have to compare, obviously, these new methods to our reference standard ultrasound. But I think these preliminary data really show that we're on our way from taking these concepts that we used to only do in 2D, combining them with sort of the latest and greatest from 3D, and creating something that's really new and unique for assessing the fetal heart. And ultimately, we hope to be able to use this to provide better diagnosis and management in congenital heart disease. So with that, I'd like to thank the wonderful group that I get to work with at the CHUV, including our clinical collaborators that are instrumental in acquiring all of this data that we then get to develop algorithms for and test some interesting ideas. And yeah, I'm welcome to any questions.
So thank you very much, Chris, for this great, complete presentation. And I forgot to mention in my introduction that he really has established himself among the world leaders of fetal cardiovascular MR. And I'm pleased to see that he was able to adopt things he's learned here in Lausanne and bring back to the fetuses. You wanted to say something?
No, just.
Okay. Now I think the floor is open for discussions, for questions. I don't know exactly where they are coming from here in this room.
Maybe you would like to put the.
If they turn on their camera, we can see them.
Sorry?
If they turn on their camera, we can see them.
Okay, so please turn on your camera if you want to ask questions. Is there even anybody online?
Yes.
Oh, at least Mary is here. Great.
Hello.
Mary, you are another expert on fetal imaging. Do you have any questions?
First of all, congratulations. Can you hear me?
Very softly.
Can you hear me better now?
Try again.
Florian did some magic. I think we can hear you now.
Can you hear me better?
Yes.
now?
Yes, hi Mary.
So first of all, congratulations. Of course, fantastic research and amazing project you have, Chris. For curiosity, I mean, I fully align with your approach where you are really going into the acquisition and the process of MRI acquisition and dealing with emotion in a more, I would say, classical way. But I was curious if in your project or if you are planning as well to explore more deep learning methods, like, okay, you got whatever you got in a fair spatial resolution, and then you are improving these later on, for instance, by learning with better quality images that might be acquired, for instance, from a preterm. So this is, I'm asking you this because this is the way we handle, for instance, the resolution problem with diffusion. We are learning from preterms, and we apply this to fetal acquisitions. So are you considering some of these approaches?
I mean, it's not something that's in our immediate plans, but I think it's really worthwhile, especially if you think that one of the strengths of this is to sort of have a framework and an acquisition system that will work for both fetal and maybe neonates and then into adolescence, which is actually just something we don't have yet. We've typically done fetal cardiac imaging in a completely different way than we do adult cardiac imaging. So I think in order to build something that would be useful for a deep learning approach, we need to kind of bridge the gap in the way that we collect these images. So I think long term, if we can, have a more similar protocol, both sort of prenatally and postnatally, that would definitely work. On the other hand, as we figure out the boundaries of this 3D imaging, it still could be that there's still a place for the 2D individual slices that we can get of really high quality, and maybe that could then inform the 3D acquisition. So the 3D acquisition is more about coverage, but maybe the 2D informs the individual quality of the images. I don't know if I'm looking at the right person. I'm looking at you on the screen, Mary.
Just
But
in the front.
oh, fair
The
enough.
camera right there in the corner.
Okay.
That's where the camera is. So.
Sorry, I was just watching Mary from the side. So.
Thank you, Chris. Thank you. I think it
No
makes
problem.
sense. Yes.
Okay.
Thank you. Are there any other questions?
If maybe those who are on the other side of the camera to put on your cameras, it would be great to see, for us to see as well who was there.
Still raised hand. So maybe in the meantime, Chris, I mean, echo is the first stop, right? And so in which cases do you see MR adds above and beyond of, you touched upon it already a little bit, adds above and beyond of what echo is capable of doing, and why should this be disseminated more broadly worldwide once this technique works? Maybe once we add Mary's super resolution techniques, etc., etc. So why go to the next step and do MR and not just stay with echo?
So I can speak both to sort of the technicalities, but also my own anecdotal experience. So after I finished my PhD, I stayed in Toronto for about 6 months, and that was really the question, and the reason was to provide MR in cases where the echo was inconclusive. And so the most common instances of this I talked about a little bit earlier on. It's if you have something that's blocking your acoustic window to the fetal heart. So again, this can be obesity, it can be low amniotic fluid, or just the fact that the fetal bones are preventing you from getting a really good window. And now, of course, as ultrasound technology improves, maybe this limitation becomes less of a problem. But at least right now, these are instances where we really found clinical use for MR just from purely to answer the question we would have with ultrasound, but couldn't because of this limitation. Going further from that, though, if you think of the way that MRI quantifies something like flow, we can get these pixel-wise quantitative maps of flow throughout the heart, which, if we can do this in the fetus, and there are some methods developed by some groups that are already getting us closer to that, it takes us beyond just examining whether the structures are connected correctly. It also allows us to interrogate things like, what is the impact of those structures being incorrectly connected? Does a narrowing of a vessel actually mean that not enough blood flow is getting to the brain? Or in these borderline cases, can we provide more information? And so it's really twofold. It's answering the question that ultrasound can't at the time because they can't see the heart well enough, but also eventually adding things that MRI does really well, which is the reason why we use MRI in congenitals for pediatrics and adults. I think that's the basic answer.
Thank you very much. Yes, please.
Yeah, it's just to follow up on this question. So when you clinically, if you can't have the information that you want from ultrasound, is that already an indication to go to an MRI, to do an MRI in the clinic? So
there's a couple centers now that I'm aware of, at least, that really is now being established as their clinical pathway. It's this is, if you think of this field's barely 10 years old. Just being able to physically do cardiac MRI in a fetus is about 10 years old. So obviously different countries, different institutions have to create all of the logistics with insurance and within the services to have a clinical indication. Otherwise, the majority of it's been done for research so far. But I know of at least 2 big centers where that now is the case. They have an inconclusive ultrasound and they can go to MR to try and complement the diagnosis.
Any other questions while they are still thinking online? I've learned in my career that scarcity creates opportunity. And I've learned that the hard way when I first started scanning at 3 Tesla. Now, 3 Tesla, people think, oh, bigger signal to noise, everything is hunky-dory. No, it isn't. It is just a heck of a lot of technical problems that have to be solved if you go to higher field strength. However, solving these problems then led to technology, to methods, to hardware, to software which kind of bubbled down, so that we could improve, 1.5 Tesla imaging. Now here, we also create scarcity in that we have to image a smaller object that beats faster, where we have complex situations like those mentioned by you. Do you also see then from, when we learn from fetal imaging, what could bubble up to a congenital adolescent imaging or adult imaging because of all the technology that had to be developed for addressing these problems for the fetal heart?
Well, I mean, one very good example is, this pediatric imaging. The way I kind of showed the narrative a little bit backwards, where I sort of show that the pediatric imaging informs an idea for fetal. Really, these things were happening in parallel in my brain. I thought we could get here with fetal, but it made a lot more sense to try and look at these cases of pediatrics. And so both of these techniques are still in their development. And so as we, there's questions we need to ask. Like, for example, in these real-time images that you see here on the left, these are reconstructed about every half second to a second. That might not be fast enough for the fetus. So we're gonna really push our reconstruction algorithms and test new ideas to try and give this the best frame rate as possible, which in turn would benefit this pediatric imaging, as one example. Another example is typically in adult and pediatric imaging, you can see here that the field of view that we have, we just try and get as tight to the heart as possible without having wraparound artifact from not having a big enough field of view. And that seems to work reasonably well. If we do go to an even bigger field of view, with this type of imaging, we start to have some artifacts along the periphery of the field of view. We have to deal with things like whether we want to be selective in our excitation. So that means, do we just want to image a certain part of the volume and try and cut off things that are above and below it? Or do we want to just excite the whole volume and then deal with the fact that we might have some artifact? For the fetus, this is going to be a real challenge because we want a very big field of view. We really want to capture the whole fetus. And so I think this is going to make us revisit some of the ways that we set up this protocol, which again, in turn, will wrap back to adult and pediatric imaging. If we can come up with ways to really either deal with the artifacts that we see at the edge of the field, or decide to invest time into a really good selective excitation, this is going to trickle back to the other parts. So it is very much a case of something that needs unique solutions because it's very challenging, but then it could benefit other types of imaging as well.
Other questions? Yes, please.
Yeah, I'm not very much in this field, but I'm just fascinated by what you've been telling. And I know, so ultrasound has been used in the traditionally for imaging. And I imagine that, practically speaking, when clinicians start to work with now the MRI images as well, that they will have an extra tool to integrate and to understand if there is any problem. How difficult or easy is it for you to integrate both images in a final kind of maybe assessment? Is that something that you can?
Yeah, I mean, that's a great. Sorry. It's not something that we've really assessed. Sorry, I've got to go really way back. There we go. So it's not really something we've assessed in terms of trying to fuse these things together yet, but it's a really interesting idea, especially with the ultimate goal of being complementary, if maybe one modality provides something that the other can't, much in the way we fuse, say, MR and PET together or something like this. So I think it's worth investigating. Obviously, the challenges are going to be things like we have very different contrast. Can we get the scan planes well aligned? But going to 3D and essentially having all the scan planes, we might be able to retrospectively interrogate and find the same positions that we do with ultrasound and try and complement them in this way. So it's a really interesting idea.
I guess it's not an easy task.
No, but none of this is. And that's why it's good research. And if there's a
perfect candidate, then
we'll see.
Thank you.
Yes, please.
It's very interesting, this fetal imaging. I was wondering, to compete with ultrasounds, since we already have a ultra low field magnet MR scanner. So what is challenge to implement this, for example, ultra low field MRI? Because this will be, in terms of cost, everything will be competing with the ultrasounds, and it can provide more actually quantitative assessment from MR point of view. Then it can kind of more widely spread in the clinics. You know, how is the what you see the perspective for this application in the ultra low field?
So I have a couple perspectives. One, I'm very excited that we're getting one of these magnets. This is, of course, you're absolutely right. It's a great application. That you can't ignore the cost differences between doing an ultrasound exam or an MR exam. And so anything that we can do to be able to do this type of imaging on a scanner that will eventually be more accessible and more used, I think is really key. It creates an additional technical hurdle that I think all of us are going to face trying to do cardiac imaging at low field, which is lower SNR, different hardware that's maybe not as good as what we're used to. I mean, we're very lucky at the CHUV. We have really top-notch scanners that we do our cardiac imaging on. But again, they're great research challenges. And so being able to marry something that's clinically very useful, and especially in that case, something that could be more widely disseminated, I think it's awesome. It's a good engineering challenge. I'm sure it's going to inspire more projects and more grant writing down the road. But maybe just additionally, just quickly I can talk a little bit on my experience of how the consideration of cost for MR and ultrasound has been dealt with both at the centers I've worked at and the ones that I know from the field. It's really, and I think it'll still be this way for a while, this is something that will be done at large centers, right? It's not intended to be sort of a screening tool like an ultrasound would be in smaller hospitals. But where it still has real importance is you have many areas in the world. And so if I go back to where I was in Toronto in Canada, the Hospital for Sick Children is a huge children's hospital, but it has to accommodate a really, after Toronto, a very dense, sparsely populated area. So you have people that come from far away and they come to this hospital. And if there's a suspicion of a congenital heart defect, it's really important that the clinicians know, can we send this pregnant woman back to their smaller hospital, or do they need to be here for an intervention? And so that, I think, cost benefit equation there is then satisfy that you really need to have this type of imaging in these large centers to make sure that you have the right solution. Now, maybe someday we'll also have MRs in these, low field, low cost MRs in these smaller centers too. But then in that instance, it'll also still be a question about having all the information there for what I'm sure by that time will be an even growing list of interventions that we can do. So again, this field of fetal cardiac MRI is quite young, but relatively speaking, interventions during pregnancy for cardiac abnormalities is quite young as well. So whether it's drugs, whether it's surgery, as these things improve, I think we'll see the same thing we see in all imaging, which is new treatments require better imaging to make sure that we do these things in the appropriate way. So yeah, basically low field, very exciting, but there's still use for the MR, even if it costs more than ultrasound.
But again, I would say scarcity creates opportunity, right?
Exactly.
And the scarcity here is the signal to noise. So,
yeah.
With Mary's techniques, super resolution, AI guided or assisted reconstructions, etc., I think this will go a long way. You mentioned the difficulties, and I think the difficulties are not only of a technical nature, they may be of an ethical nature if you think about certain patient groups, but they may also be of political and geographical nature, right? So it's actually a very, very interesting problem if you think about it. And if you think about the geographical aspect, us being here in Lausanne and the children's hospital being built, to our annoyance, because it makes a lot of noise every day, right next to us, it's pounding, it creates a headache. But they're building a children's hospital, and into that children's hospital they will put an MRI scanner. So what is your projection into the future? How these two things, here we have this great opportunity, this fetal imaging. On the other hand, we have this great opportunity in the children's hospital with an MRI scanner where they might benefit from it. So how are you planning of embedding this into this political geographical landscape, ultimately to the benefit of the patient?
That's a great question. I think it can be equally challenging navigating those things, but luckily I've now had the experience of you can't do this research unless you develop relationships with clinicians, with the support staff at the hospital. So ultimately, I think what it comes down to is really, as much as these are fun engineering challenges, they really require frequent meetings with clinicians to understand, is this moving towards something that they will really want to use, or are we just doing it for the sake of an engineering challenge? And so my plan is really to continue to address the technical side, but also coming back to this idea of having something that really bridges the gap between what we can do prenatally and postnatally. I think that's a logical fit for a scanner that's embedded in a pediatrics ward. Being able to do everything there pre and postnatal, I think, will be really important. So it's a little bit of, hopefully the results speak for themselves, but at the same time, they can only speak for themselves if you're building something that clinicians are really going to be invested in.
Yes, final question.
Yeah, are there special restrictions for putting children in an MRI scanner?
For children in the MRI scanner?
Yes.
Not really restrictions. There's always safety precautions in the MRI, in any MRI, and so the considerations being acoustic noise, so it's loud. The changing magnetic fields are loud, so ear protection is important. And then there's always the possibility of tissue heating. So this all operates in the RF spectrum, and so. But generally, there's been a lot of studies in fetal imaging as well. We, of course, wouldn't do this without sort of these landmark studies that have looked invasively in animal models and quantified the amount of potential heating and set us very conservative limits that we still stay well under in terms of both acoustic noise and heating.
So postnatally, you could follow a fetus right away after birth in the scanner again.
Yeah, so there was a project in Toronto that I wasn't a part of, but colleagues were doing, where that was very much the intention was to try and target the best time for an intervention in transposition of the great arteries. So when you have, it's actually one of the examples I showed, when you have the aorta and the pulmonary arteries swapped on the ventricle they're connected to, you can do a surgical repair almost immediately after birth. But because these things also often come with a premature birth, it's not always clear when this operation will be most effective. And so they were really doing serial scanning before birth, right, up until after, to the intervention and post the intervention.
All right, with that, my final word would be fetal MRI, I think, is in its infancy, and we are looking forward to seeing this grow, thanks to your work, into an adolescent. Thank you very much for being here.